Welcome to the UC San Diego Shiley-Marcos Alzheimer's Disease Research Center Brain Blast series. My name is James Brewer, and I'm the director of the UC San Diego Department of Neurosciences and the Shiley-Marcos Alzheimer's Disease Research Center. I'm a professor of neurology, a clinical neurologist, and a brain imaging researcher. I'm pleased to present to you our virtual Brain Blast series. Each of the talks included in the series have been created by Shiley-Marcos ADRC colleagues with expertise in brain aging research that focuses on prevention, detection, diagnosis, and treatment of Alzheimer's disease and related dementias. Each of the experts featured in the series studies a unique aspect of brain aging and has developed a brief overview of their current research to highlight the state-of-the-art work that's going on here at UCSD and our center. We hope you'll take advantage of this free resource to learn more about the advances in brain aging, the importance of research, and the multitude of opportunities to get involved and participate in research studies.
I'm Dr. Douglas Galasco, a professor in the Department of Neurosciences at UC San Diego. It's my pleasure to tell you about our longitudinal research program at the Shiley-Marcos Alzheimer's Disease Research Center at UCSD. First, a little background. The UCSD Alzheimer's Disease Research Center, or ADRC, is one of the original 5 Alzheimer's disease research centers that was established by the National Institute on Aging in 1984. The ADRC program was set up to translate research advances into improved diagnosis and care for people with Alzheimer's disease. And the ultimate goal was to help to find ways to treat and ultimately prevent Alzheimer's disease. There are now over 30 ADRCs across the USA. The scope of our research includes not only Alzheimer's, but a number of conditions that we classify as related disorders. These include Lewy body dementia and frontotemporal degeneration, as well as vascular cognitive impairment. And we also are extremely interested in changes in cognition that happen over aging. Our research mission is firstly to carry out research into the causes, treatment, and prevention of Alzheimer's disease and related dementias. The ADRC acts as a local and national resource by providing research subjects and materials we collect from those subjects to researchers. We provide training opportunities for clinicians and researchers to help to develop their interests and careers. We contribute to educating healthcare professionals and people in the community regarding Alzheimer's disease. And we provide outreach to the diverse San Diego community regarding the diagnosis, treatment, and care of people with Alzheimer's disease. We have several themes that drive our research. Firstly, we're extremely interested in mechanisms of disease. It turns out that Alzheimer's disease is a complex disorder, and a number of different factors and mechanisms contribute. Risk factors can broadly be grouped into aging, genetics, and environment. Environment is an enormous risk factor all on its own, and it broadly includes social and medical factors, as well as things people are exposed to. These factors interact across one's lifespan and influence good and bad brain aging, and ultimately the risk of developing Alzheimer's disease and related disorders. In order to study how risk and cognition change over time, it requires long-term follow-up through the ADRC. This part of our mission is critical because as we understand mechanisms of Alzheimer's disease, this will enable us to develop interventions that improve outcomes and ultimately result in effective treatment. If we think of healthy brain aging as a balancing act, there are things one might do that improve resilience or reserve, such as cognitive activity, social activity, exercise, and sleep. And there are things we need to be concerned about that may contribute to age-related damage, such as high blood pressure, diabetes, diet, and stress. This is a simplification of some of the factors that contribute to this balance, and this plays an important part of our research and research assessments. Another one of our research themes is markers and maps. What motivates this is that Alzheimer's changes in the brain begin a decade or longer before the very first symptoms. We are able to measure changes, including the buildup of pathology in the brain while people are still alive. In order to do this, we use tests that are called biomarkers to construct a map of change over time. Biomarkers can measure things such as brain structure. One example would be MRI, brain chemistry through things like PET scans or sampling cerebrospinal fluid or measuring things in the blood, and finally brain function, for which our primary measure is cognitive testing. This diagram shows how brain cognition and brain structure and function may change to influence cognition and how somebody functions in daily living. The ultimate objective is to preserve normal brain aging and normal brain function. And some people are going to start out that way and over time will progress through changes that put them on a pathway to develop Alzheimer's disease. Through building a detailed map, we know that Alzheimer's undergoes several different stages. The earliest stage involves the buildup of pathology while people have no symptoms at all, and this is called preclinical Alzheimer's disease. An intermediate stage is called mild cognitive impairment, where people have mild changes in memory but are largely still able to function in daily life. And then further downstream is the development of dementia, where the cognitive changes result in impairment of function. Underneath the surface is the development of brain pathology with the buildup of plaques and tangles. Plaques are made of amyloid and tangles are made of tau, and these were the structures that Professor Alzheimer first described in 1907. And then importantly, factors such as vascular disease are also building up. So is going to take all of these into account. In order to accomplish all of this, we need research participants. And in order to really understand the interplay of different factors in relation to brain health and disease, we aim to enroll and follow a diverse group of volunteers. Diversity includes age, sex, we're obviously interested in enrolling both men and women, ethnic background, we have a particular interest in Latinos in San Diego, and diversity of risk factors. We're interested in Alzheimer's and related disorders, as I mentioned before, and we also are following people who are 65 and older who do not have memory problems. Participants in the ADRC come in for annual detailed visits and are also offered the ability to take part in additional studies through affiliated researchers. It is critical to be able to compare all of this data that we collect during life with changes in the brain that occur that we classify as pathology, and therefore the ADRC supports a program of brain donation after death among our study participants. People who come in for our study undergo a variety of procedures. These include neuropsychological testing, so pencil and paper tests of memory and other cognitive function. We draw blood. We perform a lumbar puncture on people who consent. We perform research MRIs, PET scans. A skin biopsy is taken to be able to produce things called induced pluripotent stem cells, which are ways of growing nerve cells out of people's own skin. And then, as I mentioned before, we support an autopsy program. So as I mentioned, all of this cannot happen without people wanting to be part of our exciting and diverse studies. So we would like you all to find out more about how you can help to contribute to our research. And this slide lists a number of ways of contacting us and finding out further information through telephone, email, the internet, or our Facebook page. Thank you very much for watching.
Hello, everybody. My name is Erin Sunderman. I'm an assistant professor in the Department of Psychiatry at University of California, San Diego. And I'm here to tell you today about a new study we are conducting through the Alzheimer's Disease Research Center that will examine the potential of natural language as a marker of early stage Alzheimer's disease. A major advantage of this study during the social distancing times is that it is a short one-time study visit that can be done completely remotely from the comforts of home. So first, I'd like to tell you what we know so far about the effects of Alzheimer's disease on language. We know that our ability to speak clearly and identify the words that we need to communicate becomes more difficult in the early stages of Alzheimer's. These problems with word finding can result in changes to the timing, acoustic, and linguistic elements of speech, including the number and length of the pauses we take, the number of total words, the structure of our sentences, and our pitch. These alterations to our speech may provide an early marker of underlying and subtle changes in our thinking skills and memory, and possibly even before more formal clinical memory tests. However, there's still a lot we don't know about the effects of Alzheimer's disease on language. So for instance, which elements of speech are most affected by early Alzheimer's related brain changes? And how do these elements of speech relate to biological markers of Alzheimer's that we collect in life, such as certain protein levels in cerebrospinal fluid and through brain imaging techniques? Lastly, are speech elements from our spontaneous or free speech versus our speech when we're reading aloud scripted texts more sensitive to changes in thinking skills and memory? All these questions we hope to answer in this study. So participation in this study involves a brief one-time video call over Zoom with an ADRC staff member that will take about 15 minutes. After consenting to study procedures, you'll be asked to talk for 5 minutes about a memorable event that occurred in your life in early adulthood in as much detail as you can recall. This can be any event at all that you remember well, including a wedding, first day of a new job, birth of a baby, moving into a new home, etc. Then you'll be asked to read aloud 5 paragraphs of text that will appear on your computer screen one by one. These tasks will be recorded, and the recordings will be run through a computer program that will measure all different kinds of speech elements. We'll then be able to examine how these measures of different speech elements relate to your performance on the battery of thinking tests that you complete during the standard ADRC visit. And for those of you who had a lumbar puncture done at the ADRC, we'll be able to look at how these speech elements relate to Alzheimer's related markers in cerebrospinal fluid. So we will welcome anyone into the study who participates in the annual study visits of the ADRC, is fluent in English, and has access to a computer screen, and who's not in the more advanced dementia stages, since the ability to participate would be compromised in this stage. We think this study is important for multiple reasons, but first of all, there is a pressing need in the field to identify early markers or signals of potential Alzheimer's disease, because the earlier we can intervene, the better the chances that our currently available treatments and interventions can have a positive impact. It's possible that alterations in speech may be an even earlier marker than our formal tests of memory. And also, unlike many of our other methods of detecting early brain changes, measuring natural speech represents a non-invasive, low-stress measure that can be done easily and inexpensively at a clinic visit. So thank you for listening, and we welcome anyone who's willing to participate. As always, we are looking for people to devote their time and effort into Alzheimer's research, and it is that generous devotion to time and energy in Alzheimer's research that really drives the fight against Alzheimer's disease. So thank you.
Hi, my name is Dr. Christine Smith. I'm a research health scientist at the San Diego VA and also an assistant professor of psychiatry at UCSD. And I'm running a research study to develop a new cognitive test to help identify Alzheimer's disease earlier and better than traditional methods. By the time people are diagnosed with Alzheimer's disease, major changes have happened in the brain, and we need more sensitive tests to help identify people at risk better and earlier before a major brain injury has occurred. Individuals with mild cognitive impairment are thought to be at a higher risk of developing Alzheimer's disease. These individuals have mild difficulty learning new information, but can have severe difficulty remembering information from the past. Currently, individuals are assessed using traditional methods such as standardized neuropsychological tests and overall clinical evaluation. These traditional methods are used to determine whether an individual has mild cognitive impairment or Alzheimer's disease. However, these traditional tests do not measure memory for the recent or remote past. Therefore, we could be missing some cognitive impairments. Previous research has provided evidence that tests of notable news events are useful tools for measuring memory for the past. It's our goal to use a news events test to determine if it can improve identification of individuals with Alzheimer's disease before major brain injury has occurred. Our study addresses the following questions. Can a news events test aid in identification of those at risk for Alzheimer's disease? What brain regions support memory for the past? And are these the same brain regions that decline in Alzheimer's disease? For our study, participants complete traditional neuropsychological tests, and then they complete 2 MRIs, one measuring brain structure and one measuring brain function during a news events test. They're also asked to complete a medical history form, some questionnaires, and provide a saliva sample. We have very flexible schedules, and you can come in for the 3 study visits according to your schedule, even on evenings or weekends. Participants who are interested in getting involved in the study must meet our inclusion criteria. So they must be currently experiencing mild or moderate memory difficulties, be 65 years or older, right-handed, have lived in the US for most of their lives, most of their adult lives, and be fluent in English. Participants must also not have any of the following conditions. They must not be diagnosed with dementia or Parkinson's disease, have a pacemaker, be left-handed, and have any current major psychiatric or neurological conditions, as well as a few other things to make sure that they're good candidates for the study. From our preliminary data and previous research, we anticipate that the brain regions supporting performance on the news events test are the same brain regions that decline in Alzheimer's disease. Therefore, we anticipate that our news events test will help improve identification of those at risk for Alzheimer's disease better and earlier than existing tests. Thanks for listening.
Hi, everyone. I'm Dr. Rayanne Moore. I'm a clinical neuropsychologist and an associate professor of psychiatry here at UCSD. I'm going to be talking today about a virtual reality tool for measuring thinking skills. Alzheimer's disease impacts multiple areas of brain function, including thinking skills such as the ability to remember, balance, as well as sensory abilities such as hearing loss. The brain and thinking changes with Alzheimer's disease start small, and they can be hard to detect in the early stages of the disease process. There's a need for improved screening tools to assess brain changes associated with early Alzheimer's disease in order to intervene early when our current treatment options are most effective. There's also a need for improved screening tools for early assessment, because if we can intervene early, that will give people the opportunity to maintain quality of life for as long as possible. So together with Emory University and other colleagues at the Georgia Tech Research Institute, we have developed a virtual reality tool to measure thinking skills and balance. This new and exciting tool, we're hoping, can detect early signs of thinking changes and balance problems in people before they progress to seriously impact daily life. If found to be a reliable way to measure thinking skills and balance, our virtual reality tool could move testing out of the clinic and into the waiting room. And we're hoping ultimately that this tool could be something that could be used in the home, and that people could use it just as they used to check their blood pressure. You're just checking your thinking skills regularly using our virtual reality device. Ultimately, we're hoping this could eliminate many of the burdensome features that are associated with traditional cognitive testing, including long, burdensome assessment batteries, often upwards of 6 hours or so, the required space and trained staff, and the expense of having to conduct these traditional assessments. Our virtual reality testing involves sitting in a chair, so it's done completely seated. You'd wear a headset on your face and respond using a little handheld controller, as well as respond with different head movements. Our tool was designed to briefly and accurately measure memory, reaction time, and other thinking skills, as well as to measure non-postural balance. So that is, we found a way to actually measure balance based just on how you move your head, like this, back and forth, so that the balance test can be done while seated and doesn't pose a risk for falling. Our study is for 2 years in total, so if you agree to participate, we're hoping you commit to participate for 2 years. It involves 5 different study visits. These visits are 6 months apart and alternate between a full visit and then very brief visits. So you would start out with a full visit. That would include coming to our laboratory, taking our traditional paper and pencil-based cognitive tasks, taking our virtual reality measurement tool, having a medical interview and a blood draw. This would also involve going to the San Diego State University audiology lab for a standard hearing test. And then you would come back at 6 months for a very brief, less than an hour visit, and just complete our virtual reality measurement tool at that time. At 12 months, you'd come back for a full visit again. And then at 18 months, just the virtual reality. And then at 24 months, the full visit one more time. We are flexible in terms of scheduling, and we can accommodate Saturday schedules if a Monday through Friday timeframe doesn't work for you. So what can we learn with virtual reality? Well, we're hoping that our tool will be a way to really measure thinking abilities that are similar to paper and pencil tests, and will be as accurate and reliable as our paper and pencil tests, so they could ultimately move testing out of the clinic and into the real world. We're also hoping it'll be a way to detect minor brain changes associated with early Alzheimer's disease, and that it will ultimately, hopefully, help people be able to identify changes in cognition and balance over time. So if you're interested in participating, we do have some inclusion criteria, which includes being fluent in English, age 60 years of age or older. You can be a current Alzheimer's Disease Research Center participant or a new participant to the center. Currently not diagnosed with Alzheimer's disease or an Alzheimer's disease-related dementia. And we do have some other inclusion/exclusion criteria that we would screen for over the phone prior to your enrollment in the study. So thank you for your attention today, and we're really hoping you'll reach out and participate in research at the ADRC. Thank you.
Hello everyone, my name is Alejandra Moreno. I'm a postdoctoral fellow at UCSD. I work in the neuroscience department, and the study that I'm going to be sharing with all of you today is called Perceptions and Attitudes of Aging-Related Research Among Older Latinos. And before we begin, I just want to share with you some information about Alzheimer's disease. Alzheimer's disease is the most common cause of dementia. Dementia is a general term, and there are different types of dementia, Alzheimer's being one of them. Symptoms of Alzheimer's vary from person to person. However, memory problems are typically one of the first signs of cognitive impairment related to Alzheimer's disease. But there's also a number of other problems with language, mood changes, and unpredictable behavior. Currently, Alzheimer's disease is the 8th leading cause of death among Latinos here in the US. And this number is likely underreported due to the stigma that exists in this population, low levels of knowledge, awareness about Alzheimer's disease, and misdiagnosis as well. Currently, while research is beginning to uncover this impact of Alzheimer's disease among Latinos, what we have learned to date is that Latinos may be at greater risk of developing Alzheimer's disease than non-Latino whites. And this number of Latinos, as you can see from this graph, is only expected to increase to 3.5 million by the year 2060. And despite this growing impact of the disease of Alzheimer's disease in the Latino community, only 1% of Latinos participate in studies. So this reflects an urgent need for Latinos' participation in this type of research. Okay, so now I want to share a little bit more about the study that I'm conducting. So the purpose of this study is to understand the attitudes and perceptions of older Latinos as it relates to Alzheimer's disease research. In order to participate, you have to self-identify as Latino or Hispanic, be 50 years or older, be able to communicate in either English or Spanish, and live here in the US. This study consists of a single phone call with me, which takes about 1 hour to complete. During this phone call, we're going to be discussing Alzheimer's disease research, your willingness to participate in this type of studies. And it's basically just a conversation. And at the end of the study, you will be compensated with $40 for your time. And the reason that we are doing this study is because we want to be able to better understand what older Latinos think and understand of Alzheimer's disease research, as this is an important step to increase their representation in all realms of Alzheimer's disease research. But most importantly, we want to address the impact of this disease in the community. We want to raise public awareness about Alzheimer's disease. And based on the information from this study, we want to develop guidelines in Spanish and in English for research personnel, for people working in the community, in order to be able to better discuss this type of research with older Latinos. And if you're interested in participating, you self-identify as Latino or 50 years or older, please, you can give us a call to the phone number on the screen, 858-215-2260. Leave us a message. Let us know that you're interested in participating, and we'll be giving you a call. The ADRC at UCSD is currently looking for volunteers. So if you want to hear more about any of these opportunities, please feel free to visit any of our websites for more information. And we're looking forward to hear more from you. Thank you.
Hi, everyone. I'm Irene Litman. I am the director of the University of California, San Diego, Parkinson's and Other Movement Disorder Center. I am a professor in neurology. I am at the Department of Neuroscience. I'm part of the Parkinson's and Other Movement Disorder Center. This is a center in which we study problems of movement, either less or more movement, and there are diseases that are neurodegenerative, such as Lewy body disease or dementia with Lewy bodies. So what are the dementia with Lewy bodies diseases? These are a spectrum of dementias that do have several clinical presentations and may have different lesions in the brain. The symptoms include problems with the behavior, with the cognition, with the movement, blood pressure, urine, such as incontinence, and sleep, that I'll discuss in a minute. There could be lesions that correspond, when we look at the brain, to Parkinson's disease and dementia, dementia with Lewy bodies, or other diseases that do associate very frequently and make the diagnosis even more difficult. That is Alzheimer's disease, for example, or vascular problems. So this makes the diagnosis quite difficult. And because it is so difficult, we need to find ways of diagnosing these diseases so much earlier so we can intervene. The Lewy bodies are lesions in the brain that are shown like this when we look with a microscope, and we can see these inclusions inside those cells. So all these diseases have these Lewy bodies that do have a protein that it doesn't matter the name, but it's called alpha-synuclein. And the important thing about this is that nowadays there are starting to be therapies to try to slow this disease or these diseases. So if we were to know to diagnose these diseases much better, we could have all our patients into studies that do correspond to therapies that could even improve the symptoms or could improve, more importantly, the progression of the illness and make it so much slowly, or perhaps even to try to cure, or not cure, but at least stop the progression. It is impossible to really call cure when we have still lesions in the brain. You may be familiar with some of these things because you may have read in the newspaper someone that we all love who had dementia, and not only dementia, but also his symptoms would change, and some days he would have much better functioning than others. Others in which he could do things, could call, could talk, and others in which he wouldn't even know how to do that. But in addition, what is at times very bothersome are the visual hallucinations. These are seeing things that are not there. So this happens as well with problems of movement, and the problems are the Parkinsonism. That is really slowness, difficulty in getting out from chairs, difficulty walking, as well as problems such as stiffness and perhaps tremors. Patients do have problems with urination. They urinate very frequently and at times do develop incontinence. Also importantly, the blood pressure, rather than increase a little bit when we stand up, it lowers, and it may lower to very, very low levels that makes not enough blood going into the brain, and the brain suffers, worsening on top of it the dementia. In addition, there are problems with sleep. One have really bad sleeps, and so enacts those dreams. REM behavior disorder, that is how we call it, may go into either Parkinson's or dementia with Lewy bodies. So this is a very important symptom that one needs to consult for. So what is our study goals? Well, one of them has been already achieved, that is to form a group of centers that are dedicated to the study of Lewy body diseases. So what we're doing is collecting detailed clinical information, collecting blood, collecting imaging such as MRIs from a group of subjects that do have dementia with Lewy bodies that I just defined, or may have mild cognitive impairment that doesn't affect the functioning of the person in the house, or that doesn't impair the functioning socially. But in addition, these patients do have the high likelihood of have dementia with Lewy bodies. And in order to have that high likelihood, what happens is that they should have either hallucinations or the enacting of the dreams or other symptoms that suggest, such as Parkinsonism, the possibility of mild cognitive impairment that will lead to dementia with Lewy bodies. Patients can also present with Parkinson's disease that eventually evolve into dementia. So who is coming to our group is going to be followed on a yearly basis. And the overall goal in the future is to collect all these elements, the bloods, the urines, the imaging, to be able to find markers of the disease, to be able to find ways in which we can diagnose the disease even before most of these symptoms occur. So we can treat patients very, very early. That is the overall goal. We can also study these patients and can find the better therapeutics that could be available. We need to do a lot of study in these patients. And what we're doing is searching for patients that do have these diseases, following them up in 10 sites in the United States on a yearly basis at 1, 2, or 3 years, and 6 months after the baseline. We do see them during weekdays, and they do have a neurologist that is specialized in movement disorders, in dementia, that evaluates them and do the cognitive testing, the questionnaires, and may have as well at times, if they wish, a lumbar puncture. We need a lot of patients at this time to make this study successful. So we need you or your family members or your patients participate in this study. And we appreciate and will be happy to help you in any way possible. We see our patients at the Altman Clinical and Translational Research Institute that is across the Thornton Hospital. Our research coordinator is Rachel. You can see her telephone and email in this slide. Thank you for your attention. It has been my privilege talking to you.
I'm Neil Swerdlow. I'm a distinguished professor of psychiatry at UCSD. And I'm going to tell you about biomarker predictors of memantine sensitivity in patients with Alzheimer's disease. The short title would be the Memantine Study, and that's the name of the study that we're doing right now. So memantine is a medication. It's also marketed under the name Namenda. It's approved by the FDA for treatment of Alzheimer's disease. It's generally very well tolerated, and it can be used in combination with other medications for Alzheimer's disease. So it's already approved and it's well tolerated. The problem is that memantine only helps some people with Alzheimer's disease, but not others, and there's no way to know in advance who will or who won't benefit from memantine. In this study, we're looking for a biomarker, a signal to tell us who will or won't benefit from memantine. Recently, we've been able to identify some signals in the brain that tell us that memantine has reached parts of the brain and is working. We're now testing whether these brain signals can serve as biomarkers to help us know in advance who will or will not benefit from memantine. And that's the purpose of this study. Here are some results from a study in 2007 showing that people with Alzheimer's disease who take memantine show some improvement, while those taking a blank or a placebo pill show worsening of symptoms. And this is over a 24-week period. So we already know and have known for some time that people with Alzheimer's disease who take memantine will show benefits, whereas those who take a placebo pill will not. But even among the group that took memantine, there were differences in sensitivity. Some people got a lot better, some people got a little better, and some people didn't get better or even got worse. So why is it important to find a biomarker for memantine sensitivity? Without a biomarker, one person might be sensitive to memantine, another person not, and we can't tell by looking at them who is or who isn't sensitive. In the study of how memantine affects, if we give memantine to everybody, only some people will get benefit. Most people may not, and there's no way to know ahead of time who will or won't benefit. With a biomarker, we can identify a person who is memantine sensitive and a person who isn't by doing a laboratory test. Then we screen ahead of time who is showing that biomarker for sensitivity and only give sensitive people memantine. In that type of a study, most people will benefit from memantine. So in the memantine study that we're doing, there are 3 phases. The first involves a screening day, which takes about 5 hours and involves tests of your memory and a physical examination. The second part of the study involves testing for those biomarkers. These are brain signals that we detect through a technique called EEG. This takes 2 full days spread apart by about a week. During those days, on one day you'll take a fake pill, a placebo, and on another day you'll take a real pill of memantine, 20 milligrams. After that, we'll do tests that measure brain activity using EEG. This involves wearing a cap that have sensors on it. Or EMG, which involves putting plastic electrodes on your cheek here to measure your eye blink response. And this is how we measure the memantine signals. After we're done with those 2 test days, we enter the treatment phase of the study. In the treatment phase, everyone gets memantine for 24 weeks at the same dose that's normally used to treat Alzheimer's disease, and that's 10 milligrams twice a day. So everybody in this study gets a full therapeutic trial with memantine. There's no placebo treatments. Participants then complete memory tests and get a brief physical exam 8, 16, and 24 weeks later. If memantine seems helpful to you, you can continue using memantine after the study ends, prescribed by your own doctor. At the end of this study, we'll know which of our participants got a little better, a lot better, or not better at all from memantine. And we'll know that by testing your memory over the course of the 24 weeks of treatment. Then we'll look back at the test results from the 2 laboratory test days and determine which, if any, of those results, those brain signals, accurately predicted the clinical response to memantine. And if we can find one of those signals that predicted those responses, we'll have a biomarker. We need volunteers for this study. It involves 2 full days of laboratory testing and then 24 weeks of treatment with the active treatment of memantine. Thanks very much.
Hi, my name is Dr. Emily Reese. I'm an assistant project scientist in the UC San Diego Neurosciences Department. And today I have the pleasure of sharing with you an exciting new project that we're starting in collaboration with the UCSD Alzheimer's Disease Research Center that will be examining the role of the blood-brain barrier in Alzheimer's disease. Now, current models of Alzheimer's disease posit that particular brain changes occur along a characteristic time course during Alzheimer's disease progression. Initially, the toxic proteins, known as amyloid and tau, accumulate around and within brain cells, and these deposits may occur a decade or more before symptoms appear. Next, structural changes occur in the brain that can be measured with the imaging technique magnetic resonance imaging, or MRI. These changes include degeneration of the white matter connective pathways, along with the death of neurons, which manifest as brain shrinkage on MRI scans. Eventually, cognitive impairment begins, with notable memory problems, and ultimately the functional and behavioral changes that are characteristic of full-blown dementia emerge. However, there's growing consensus that this model of Alzheimer's disease is incomplete, and there are critical pieces missing from this puzzle that will help to clarify how, when, and why Alzheimer's disease develops and progresses beyond just understanding which changes occur at what time points. Now, a major clue to understanding this puzzle is the fact that vascular dysfunction coexists with Alzheimer's disease in the majority of cases, estimated around 80% of Alzheimer's disease cases. Furthermore, vascular risk factors such as hypertension, diabetes, obesity, or a physically inactive lifestyle are also very strong risk factors for dementia, including Alzheimer's disease. Now, the blood-brain barrier is the primary interface between the blood vessels of the brain and the tissue of the brain. This interface is a protective membrane or sheath that surrounds the blood vessels and selectively regulates which substances can enter or are removed from the brain. Now, damage to the blood-brain barrier can allow substances such as toxins or immune or inflammatory factors to enter the brain. And conversely, it can prevent the healthy removal of detrimental substances such as those toxic proteins, amyloid, or other waste products. And together, this damage to the blood-brain barrier can create a very dangerous environment for the brain. Recent studies over the past several years have shown that the blood-brain barrier becomes leaky or increases in permeability, both with older age and in Alzheimer's disease. However, it's not yet clear how damage to the blood-brain barrier directly contributes to the pathological processes that we know contribute to dementia. So the goal of our study is to better understand the role of the blood-brain barrier in the development and the progression of Alzheimer's disease, and specifically how it relates to other established markers that we discussed, including the accumulation of the toxic proteins amyloid and tau, structural brain degeneration, and ultimately cognitive problems. So to assess these, or to address these questions, we are enrolling participants age 60 and older who are either cognitively healthy or who are experiencing mild memory problems. Participants should be safe for MRI and have no history of kidney injury. The study protocol will involve 2 separate visits to the UC San Diego campus. The first visit will involve a blood draw to make sure that you're safe and healthy for the MRI scan, along with a questionnaire about your health and lifestyle. The second visit to UCSD will involve a 1-hour MRI scan with contrast, which will allow us to both estimate blood-brain barrier integrity and to measure brain microstructure, or the small scale structure of your brain. Now, we have the great fortune of collaborating with 2 other scientists at UCSD, Dr. James Brewer and Dr. Sarah Banks, who are conducting concurrent studies using the brain imaging technique PET that will allow us to measure amyloid and tau accumulation in the brain. So while not required for participation in our study, it's highly encouraged that participants co-enroll in our studies so that we can compare measures of blood-brain barrier leakage and microstructure to measures of protein accumulation. Now, what do we hope to understand from our study? First, we hope to clarify when the blood-brain barrier begins to show damage. Does it become leaky early on before structural changes occur or symptoms appear, or does it occur later in the disease? Second, how does it relate to other pathological markers and processes? Do regions of blood-brain barrier leakage correspond with areas of structural damage? Does the blood-brain barrier become leaky in conjunction with or independent of amyloid and tau accumulation? Ultimately, we hope that this study will help to clarify how Alzheimer's disease develops and progresses, and that this information will allow clinicians to better treat individuals with Alzheimer's disease. So if you're interested in participating in our study, I encourage you to please contact the ADRC either by phone or email for more information. And with that, I'd like to thank all of the past and current ADRC study participants for your help in the fight against Alzheimer's disease.
Hi everyone, I am Erin Sunderman. I'm an assistant professor in the Department of Psychiatry at University of California, San Diego, and an ADRC investigator. I would like to tell you about a study that we are seeking volunteer participants. This study is focused on women and is called Women Inflammation and Tau Study, or WITS. It is well known that women bear the biggest burden when it comes to Alzheimer's disease. They represent 2/3 of Alzheimer's cases, and they tend to show higher levels of Alzheimer's-related brain changes compared to men, including deposition of a protein in the brain called phosphorylated tau, or p-tau. It is still unknown why this is the case, and we hope to help answer this question through this study. We know that inflammation plays a key role in Alzheimer's disease, and greater inflammation in the central nervous system has been linked to greater p-tau. There are also sex differences in inflammation, where women tend to have a stronger immune response than men, which is what leads to greater inflammation. Women also represent 80% of autoimmune disorders. So clearly there's differences in how inflammation is acting in women and men. And it is possible that inflammation is a mechanism underlying these sex differences in p-tau and Alzheimer's disease more broadly. Certain lifestyle factors like sleep and exercise have been shown to impact Alzheimer's risk, and in some cases this impact seems to be stronger in women, perhaps due to the effect of these lifestyle factors on inflammation. So this led us to think, could these lifestyle factors be key to developing personalized Alzheimer's risk reduction strategies that are tailored to women? In comes a study where we want to investigate how do levels of inflammation in the brain affect p-tau and ultimately memory performance in women. Secondly, how do biological factors like sex hormones and lifestyle factors like exercise influence levels of inflammation and ultimately p-tau and memory performance in women? Because these factors can be altered with either pharmaceutical or behavioral interventions, could these factors represent interventions that could reduce the risk of Alzheimer's disease? The lifestyle factors that we are specifically interested in include sleep, physical activity, and insulin resistance, which is related to one's diet and represents a risk factor for diabetes. The reason why investigations like this are so important is because up to 30% of Alzheimer's cases could be delayed or prevented with various lifestyle interventions. So identifying which factors might be most influential, specifically in women, is really vital. So any woman who is at least 65 years of age at a somewhat higher risk for developing Alzheimer's due to familial or genetic factors and not currently enrolled in a clinical trial are eligible for this study. Study participation will involve a screening visit that can be done remotely from the comforts of your own home. What is involved in the screening visit is pictured over in the blue box on the right and includes a study description, answering questions about your medical history, doing a quick thinking task on your computer, and collecting a saliva sample that you will mail to us for analysis of Alzheimer's-related genes. The study visits will take place on weekdays during normal visit hours, and each visit will last about 2 to 4 hours. Study procedures include a battery of thinking tests and questionnaires, MRI and a PET brain scan, a lumbar puncture, and a blood draw, and wearing a watch-sized digital devices that will measure the amount and quality of sleep and physical activity. So this provides a visual of the study layout, where after the remote screening visit, if you are eligible and enrolled, you would come to UCSD a month later for visit 1. Visit 1 will involve a battery of thinking tests and an MRI brain scan, a blood draw, and a lumbar puncture, so that we can measure levels of sex hormones and markers of inflammation in blood and cerebrospinal fluid. Then you will be sent home with the wrist-worn digital devices to wear for 1 week to measure sleep and physical activity. You would bring these devices back 1 week later at your second visit, where a PET brain scan will be done. 1 year later, participants will come back for 2 more study visits where just the MRI and PET brain scans and the battery of thinking tests will be repeated. At the end of the study, we suspect that we will find that certain inflammatory markers will relate to changes in brain levels of p-tau and memory performance over time. We also suspect that levels of sex hormones, particularly testosterone, and lifestyle factors will influence p-tau and memory performance by promoting or inhibiting inflammation. Our ultimate goal is that these findings will inform what kind of interventions will be most fruitful in preventing or delaying Alzheimer's disease for women, and why. So of course, none of this important work can be done without the generosity and selflessness of volunteers. It is really your participation in studies like this that will help us to put an end to Alzheimer's disease, and for that we are forever grateful. So thank you. If you're interested in hearing more about the study, including compensation, you can contact the UC San Diego ADRC. Thanks so much.
We hope you've enjoyed this presentation from our UC San Diego Shiley-Marcos Alzheimer's Disease Research Center Virtual Brain Blast series. Please look for other presentations in this series and share them with your communities. We hope that you'll also consider participating in brain research studies. We always have new opportunities for participation and are actively seeking individuals 65 and older without memory disorders, as well as those with a diagnosis of mild cognitive impairment or Alzheimer's disease and related dementias, such as frontotemporal dementia, Lewy body dementia, and Parkinson's dementia, as well as others. We're also proud to have a bilingual, bicultural team who conduct studies, visits in Spanish. Your participation can make a real difference for future generations. And for those who are interested and who live in San Diego, we'll be happy to contact you. Simply provide your contact information. Thanks for watching, and we'll see you next time.